HP Models, Solving the Ebola Crisis by Brianna, Kathryn, Zhuohe, Adam, and Markan. In the biological world, structure and function are joined at the hip. As such, how proteins are folded is of great importance to biochemistry, and any tool that can help predict structure is very valuable for both understanding disease and devising novel ways to fight them. HP models are one of those tools. For some quick background, entropy is simply a measure of randomness. The more random something is, the more likely it is to occur and the more entropy it has. The HP model first and foremost utilizes entropy through the hydrophobic effect. Hydrophobic molecules stick together in polar solution because water can form hydrogen bonds, a very favorable interaction with itself. It can't with hydrophobic molecules. By putting hydrophobic molecules next to one another, you minimize the hydrophobic surface area, and therefore minimize the number of hydrophobic polar interactions, which minimizes the number of hydrogen bonds you lose due to the presence of hydrophobic molecules. As such, in the HP model, we will give every hydrophobic polar interaction a free energy cost epsilon.
So as we all know, protein folding is not random. The HP model involves assigning every amino acid to one of two groups, hydrophobic or polar, based on the functional group of the molecule. The two categories are shown here. For example, glutamine is polar because of its 2 electron pairs on the oxygen that allow it to form hydrogen bonds. Valine is hydrophobic because its functional group has no free electron pairs. Water is polar since its oxygen can donate electron pairs to a hydrogen bond and its hydrogen atoms can accept them. Now consider a protein modeled by 6 amino acids. Let's say it can only fold in 3 shapes: pi-shaped, S-shaped, and G-shaped. Each amino acid could be either hydrophobic or polar. In the following slides, hydrophobic residues will be yellow dot candies and polar residues will be red dot candies.
Let's take 2 hypothetical sequences, HP, 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 and PH, P, PHP, each in the 3 shapes we just discussed. First, we need to count the number of HP interacting molecules. Then, we need to consider the number of P solvent interactions. Finally, we need to sum those interactions by giving each a free energy cost epsilon, as explained earlier. To find the probability that the protein will fold in a certain state, we will first calculate the weight of the state in question, given by the equation here, and then divide by the grand partition function, z. The grand partition function is simply the sum of the weights of every state. The end result is the larger the weight, represented in the numerator, the greater the probability of the particular configuration, pi-shape, S-shape, or G-shape. The protein conformation with the highest probability is called its native configuration, the configuration that functions biologically. Now, let's apply the HP model for our 3 configurations with an HP, HP, HP amino acid sequence. In the diagram, every dotted line represents an HP or H solvent interaction. Each of the 3 conformations has an equal number of dotted lines representing the energy. Each of the energies is 7 epsilon, resulting in the same weight for each conformation. The end result is that all 3 of these conformations have an equal probability of occurring. For our second example, let's take the PHP-PHP sequence. In this diagram, like the previous diagram, the dotted lines represent HP or H solvent interactions. However, in this sequence, we notice that the pi shape has the fewest number of HP or H solvent interactions, resulting in the lowest energy of all 3 configurations, only 2 epsilon. In this case, the pi-shaped configuration, because it has the lowest energy, will be the most likely configuration of the 3.
Now let's use this oligopeptide as an example to review what we've learned today. To apply the HP model to our guest protein, we first categorize its amino acids into either the H or P group. In this case, serine and histidine are polar ones. Next, from the residual length, we determine the number of potential arrangements. For example, 5 amino acids from these 2D compact configurations. Be careful, if the sequence is not symmetric, there will be 2 ways of ordering. So now we can count the bond numbers between HP and H solvent to find the energy, weight, and eventually the probability of each configuration. However, Solving 2D HP model problems by hand is barely enough for fighting our supervillain, the Ebola virus. In real-life applications, we need to use complex 3D HP models, often regarded as a self-avoiding random walk and cracked by powerful computers.
Now we understand the math behind the model, let's backtrack a bit and talk about different disease, Alzheimer's, before we tackle Ebola. In this diagram, a native folded protein structure is shown on the left, whereas the misfolded protein is located on the right. If we, as biomedical engineers, understood how to fold proteins, we could generate a way to refold misfolded proteins, or generate correctly folded proteins in vitro to reverse or aid some neurodegenerative disease such as Alzheimer's disease. In this disease, misfolded proteins aggregate together, creating plaques, which has been directly correlated to weakening of thought processes. This shows how it is important to know what a protein's native state is, and more importantly, all of the conditions that lead to the native state. The HP model will help us determine optimal native state to facilitate considerations of methods and environments necessary to match biologically known native state configurations. How do we use the model to fight Ebola, a disease that doesn't have anything to do with protein folding malfunction? Because the HP model allows us to simulate protein folding by evaluating a large number of different conformations and determining the most likely native state, we can also use it to create a completely new protein. So, after a weakness in the Ebola virus protein is discovered, a protein sequence can be developed using the HP model in conjunction with other computational models that could theoretically fold into an antibody that directly targets the virus. Ebola crisis solved. Thank you.